In this video, we'll be looking at vesicles in terms of membrane transport. So we've in previous videos, we've looked at active transport, we've looked at primary and secondary active transport. Now, active transport using carriers and channels, it really is a good way of getting a lot of things across that membrane. But there are limitations, and that is that some things are either have the wrong chemical, physical properties, and such as they're too large. So large, especially large polar macromolecules, are too big to fit through one of these channels or carriers. So what's the option for moving big stuff in and out of a cell? It's the use of vesicles. So what are they? They're small sacs that we can put large molecules into that are too big to travel across that membrane by any other way. Now, what's the hallmark of, or the noteworthy thing about vesicles, is that they're enclosed by a phospholipid bilayer. That sounds familiar, right? Because cell membranes are made by phospholipids. So this is what allows a vesicle to fuse with the actual cell membrane because they're made of the same thing. And remember that the cell membrane is that liquid mosaic combination of phospholipids with embedded proteins and other conjugate type molecules. So they fuse together. So we end up with fusion with or formation from the plasma membrane. And of course, vesicles can also come from membrane-bound organelles. Now, we're typically looking at an active transport process, and of course, this is going to require some ATP for the thing to occur. So let's look at the processes of bringing something in to the cell from the outside, and that we can collectively call endocytosis. So this would be fluid or molecules or even cells being taken into the cell. Now there's different flavors of this or different forms. One is phagocytosis, which more or less translates into cell devouring, cell eating. And this is where a cell will ingest large particles. Now what are some of these particles? It could be bacteria. It could be dead or damaged cells or parts of cells, for example. And here we can see a process of phagocytosis. That here is a bacterium that has been fused with a receptor on the cell membrane, and then that can stimulate the cell membrane to start to change shape. And as the cell membrane changes shape, we get this uprising area, and you can see we call these pseudopods. These pseudopodia come and slowly surround the thing I want to bring into the cell. Now, as this process goes on, these pseudopodia will actually kind of keep reaching towards each other, and as they do, eventually they're going to touch each other, and they have the ability to fuse because they're made of the same material. They're phospholipids. So a particle such as a bacterium binds the receptor. The pseudopodia extend surrounding the bacterium. The pseudopodia merge and they pinch off, and now we have a vesicle. And because this is a process of phagocytosis, we can call this a phagosome, some for body. And in this case, if this is bacteria, I don't want to just bring bacteria in and leave it hanging out in a vesicle. The vesicle with the bacteria could merge with something like a lysosome that has digestive enzymes. And once again, when they come into contact with each other, the membranes fuse. And when they fuse, it allows them to sort of become one big space. And this way, the bacteria are merged with the digestive enzymes. So that's one example of endocytosis, which is phagocytosis or cell eating. Now, how about if we want little things? So we're looking at not whole cells, not bacteria, but molecules. Or if I want to bring in some fluid into the cell, then I can use another process. And that process is called, it is an endocytosis. Remember, these are all endocytosis. It's called pinocytosis. And the fluid phase of endocytosis also, this actually translates into sort of cell drinking. And this is about cells engulfing fluid droplets from the extracellular fluid. Now, typically what we have is that the membrane has these coated pitted folds. And you can see that there are, so membrane coated pit right here, okay? And what happens is that the molecules can interact with that, and the transport vesicle forms. And as it does, it's starting to form. You get this invagination here of the membrane. And this is going to keep going until the membrane actually folds onto itself and touches, and eventually this will it'll split off from the membrane above, and then I have a vesicle that can travel. So the transport vesicle forms and pinches off into the cytosol. The transport vesicle loses its protein coat, I should say, and can fuse with things like endosomes. And once inside the endosome, the ingested particles, I should say, are either returned to the plasma membrane, maybe they could be sent to a lysosome, or they may actually exit the cell. This could be sort of a form of transcytosis, where things come in and then go out the other side of the cell. So there we have pinocytosis. And let's look at another type of endocytosis, and that is receptor-mediated, I should say, receptor-mediated endocytosis. So the name tells us a lot, that there's going to be a receptor, and mediated tells us that's going to be part of the control mechanism, and it's an endocytosis. So it's a special type of pinocytosis where receptors have a specific molecule that they're going to bind onto. This could be like a cholesterol, hormones, iron, etc. And we can see what happens is we have the membrane, and on the inside we have those protein-coated areas, and we call this protein-coated pit. And then we have receptors. So when the right material comes and binds onto it, remember, something that binds on is called a ligand. So if the right ligand binds on, this is the signal for the vesicle to form. So the ligands bind to the receptors in the pit, and the membrane starts to fold inwards. Now, it's going to keep, just like before, it's going to keep folding inwards, and the folding is going to keep happening until eventually the membranes touch each other up here and fuse, and then they can break away, and now I basically am left with a vesicle inside. So the transport vesicle forms and pinches off in the cytosol. It can pinch off and then travel into the cytosol. Okay, so the transport vesicle loses a protein coat. It fuses with an endosome. It's in there. And once inside, what can happen? Well, once inside the endosome, the ingested products here's some of the common fates. They can be returned to the plasma membrane, sent to a lysosome, or they may exit out the other side. Okay, so those are three types of endocytosis. Now, you're probably wondering, how about stuff leaving? Well, yes, we have exocytosis, and this is about large molecules exiting the cell. So it's a secretion. And in this case, we would have a transport vesicle, okay? And the transport vesicle, which was created inside the cell, it has a membrane around it, very similar to a plasma membrane. It'll move to the plasma membrane, and of course, because it's the same material, it'll fuse with the plasma membrane. And when it does, it's very much looks like the opposite of endocytosis. Exocytosis is when it fuses and then it opens up. And when it opens up, you can see this allows the material to be discharged into the extracellular fluid. Okay, so exocytosis. Now, there is a version of transport, which we call transcytosis. And this is interesting because transcytosis is endocytosis. So material is brought into the cell by endocytosis. The material is transported across the cell to the opposite side, and then it leaves by exocytosis. So, I could have the situation where, I have a number of cells that are forming some type of layer. And if a material wants to get through, it's having a hard time getting through there, and especially if it's big. So what can happen is the material can basically enter this side of the cell and form a vesicle, Well, let's make that smaller. Form a vesicle. The vesicle travels across. And so this was an endocytosis, bringing stuff in this side. Vesicle travels across. On this end, it can fuse with the membrane, and then I get a process of exocytosis where the material can leave. So trans means sort of across. So transcytosis is where we bring the molecule in through endocytosis, travels across the cell, and exits by exocytosis. Here's just a picture. This is an electron micrograph. You can actually see this is a cell undergoing exocytosis. You can see here's the vesicle, and it's fused with the cell membrane, and now it's open, and here's the material inside the vesicle, and you can see that material being discharged into the extracellular fluid. I think that's a great electron micrograph. Okay, once again, as you go through the textbook, they have some really, really good summaries. So the textbook is fabulous. It walks you through each individual part, and then it brings it back and gives you sort of a summary. So make sure you go back and to your textbook, have a look at it, rewatch these videos if you need to, and we'll end the video here.